Okay, good afternoon, everyone. We come to our third plenary lecture in this meeting, and it's for the cell section. And again, just a reminder, these plenary lectures acknowledges outstanding contributions to the discipline of the presenter. And it's my pleasure to introduce Professor Nick Talbot. Nick, just a year or so ago, was the Deputy Vice-Chancellor for Research at Exeter University. And in fact, I actually got to meet Nick because he was instrumental in setting up a research twinning program between the University of Exeter and the University of Queensland, where I'm based. But Nick moved to the Sainsbury laboratory, where he's the executive director, and as he mentioned to me at the diversity dinner, that's really enabled him to get back more into his research. His research is focused on the biology of plant diseases, and he utilises a range of cell biology, genetics, and genomic tools and approaches in his work. He's particularly interested in fungal infection-related development and understanding how fungi are able to invade plant tissue and suppress plant immunity. He's authored numerous scientific papers and was acknowledged for his work and elected as a Fellow of the Royal Society in 2014. It's my great pleasure to welcome Nick to the stage to give his address. Thank you.
Okay, thanks very much, Chris. Thanks for that introduction, and it's a great honour to be here. I'm really pleased to be at the SEB meeting and have this opportunity to talk to you about our work on understanding the cell biology of invasive growth by the rice blast pathogen. So what I'm going to do is talk to you about, first of all, what rice blast is, because I know that many of you won't be aware of this disease or won't be aware of work in the area of fungal pathogens. And then I'm going to talk about how the blast fungus infects rice tissue, and it has a specialized developmental process that it uses to do that. Then we're going to move inside the plant and talk about how the fungus is able to move within plant tissue, how it overcomes plant defenses, and some new work we've done on trying to work out how the fungus is able to traverse cell boundaries. So how does it move from one rice cell that it occupies to the next? So I'm going to talk about that as well. So, and primarily I'm going to focus on the cell biological aspects of the work we do. The disease is a really important one. So these are leaf blast symptoms on seedlings, and this is at the International Rice Research Institute in the Philippines in a disease nursery. And you can see those ellipsoid disease lesions on the surface of leaves. And the rice blast fungus will kill seedlings completely in heavy infections. But each one of those lesions can expand, and they can grow to up to a centimetre or so in length. And what will happen is this disease lesion will produce between 20 and 50,000 spores every 24 hours. The sporulation is on a circadian rhythm. So Magnaporthe will sporulate at the dew point. It sporulates at the optimum point where those spores can be dispersed by splash dispersal through dewdrops and will be able to land on adjacent rice leaves and start a cycle of infection. So you can imagine when you've got several thousand lesions on an adult rice plant, and then you magnify that by the number of rice plants in a field, and you can see how very, very quickly you can get massive spore inoculum buildups, which is why this disease is able to spread so rapidly from plant to plant and cause epidemics. But the pathology that farmers really fear is this one. This is called neck blast. So this is when the fungus will actually spread into stems, and it will spread into nodes on the stems. And when it does that, it will rot away the inflorescence that carries rice grain. So the panicle that actually carries rice grain will be lost. And this is what causes the really severe yield losses in the field. So Magnaporthe will cause 80 to 100% yield losses when there's a neck blast outbreak. And every year this disease claims enough rice to feed about 60 million people. So it's present wherever rice is grown. And rice is grown all over the world in tropical and subtropical regions, but particularly you can see in Asia, where 80% of rice is grown and consumed. But rice is responsible for a quarter of the world's calories, so about 3.5 billion people depend on rice for their subsistence, and around 200 million farmers depend on rice for their livelihoods. So this is a tremendously important crop, and around 30% of the annual rice harvest each year is lost to rice blast. So rice blast is present really wherever rice is grown. In every part of the world where rice is grown, there's now rice blast present. And the disease is controlled by a combination of fungicide application and the development of new resistance. But new resistance only generally lasts around 1 to 2 growing seasons before it's broken by the pathogen, because so much of it's dependent on single gene resistance, so single immune receptors, which the fungus can overcome by a simple mutation of an effector that's recognized by that immune receptor. So this is a serious and recurrent and continual problem in terms of rice cultivation. But the fungus that actually causes rice blast is capable of infecting over 50 different species of grass. So there are forms of the fungus which will grow specifically on different grass species, and occasionally they can undergo host jumps, and that happened relatively recently. In South America, there was a strain of the blast fungus which infected perennial ryegrass, so a forage grass, so a lolium species, and it made a host jump and was able to infect wheat. And wheat blast first emerged in 1985 and spread into wheat-growing parts of Brazil. And it spread into some adjacent countries, and particularly for example, in Bolivia, and in parts of Argentina, there is wheat blast. But it's been constrained largely by climatic conditions because it needs a heavy dew point, it needs very humid, warm conditions to spread. So it was relatively climatically contained within South America, until 2016, when this disease just emerged completely independently, as it seemed, in Bangladesh. And there it caused absolute devastation. It destroyed 15,000 hectares of wheat in 2016 in 8 districts of Bangladesh. Since then, it spread to 20 districts. It spread into all the bordering regions with India. And there are reports of it actually spreading into Indian wheat cultivation as well. So wheat blast is now a tremendously devastating disease. You can see it's quite different than rice blast in its symptoms. It causes this bleaching symptoms on spikes. You get sporulation at the rachis, and then there's complete bleaching and loss of wheat grain set. And it causes, again, 80 to 100% yield loss. So this disease has got a tremendous capacity to cause surprises to us. And wheat blast is a tremendous surprise because we now know that it was probably a shipment of contaminated grain, which ended up being sown in farmers' fields in Bangladesh, which had moved from South America to Brazil, to Bangladesh. So an example of sort of the problems are caused by globalization. Disease is spreading very rapidly. We're part of a project now to try and respond to the wheat blast outbreak. We're actually using CRISPR-Cas9 genome editing to try and manipulate wheat to manipulate dominant susceptibility factor encoding genes to see if we can actually engineer some tolerance to the disease more rapidly than introgression of disease resistance genes. And we're part of that program funded by the Global Challenges Research Fund. And I was in Bangladesh in February, where we're carrying out some work, because this is a really serious problem. The problem is that there's huge wheat cultivation in India and in China. And for the first time in evolutionary history, we now have wheat blast and rice blast growing right next to each other. So literally in adjacent fields. And this is the same species, albeit constrained normally by host specificity determinants, but they will intercross in the lab. So we've got the potential of interspecific hybrids forming, which is tremendously worrying. And of course, this is where most of the people of the world are. So when we talk about global food security, it's a very sobering working on this disease, both rice blast and wheat blast, because we can't really have global food security unless we deal with problems such as the outbreak of rice blasts and wheat blasts. So it's a very sobering experience working on this disease, and really one of the main motivations for my work on it. But what I'm going to talk about for the rest of the talk is really just about the cell biology of infection. And what I hope I'll leave you with is a feeling that this is, in a sense, no less remarkable than the feats that this disease has and its devastating capacity. The thing that attracted me most to work on rice blast initially was the fact that first I could carry out genetic analysis. It was a genetically tractable system. But second, it had some remarkable developmental biological transitions. So when the fungus infects rice, it infects using these three-celled spores. They stick themselves tightly to the rice leaf surface. They germinate, send out a polarized germ tube. So we've got a polarized growth form. Then it will switch into this isotropic, radially symmetrical cell called an aprosorium. This generates enormous pressure, so 8 megapascals of pressure, 80 bars of pressure, so huge pressure is generated inside an aprosorium. It sticks very tightly to the surface, and that pressure enables it to generate protrusive force, and it ruptures, physically breaks the leaf cuticle and sends out a polarized penetration, rigid penetration peg to rupture the cuticle and the epidermal layer, and then the fungus will then occupy plant tissue and rapidly fill plant cells and then move from cell to cell until it ruptures again the cuticle to form these aerial kinidiophores and sporulates again. So you can see that the whole life cycle is punctuated by these transitions between polarized and non-polarized growth. And it was really trying to understand that attracted me to work on rice blast. Okay, so how does the fungus then infect host plants? How does this specialized cell, the aprosorium, form, and then how does it function? So this is what an aprosorium looks like. This is a scanning EM of an aprosorium. So this is a cell which is about 8 micrometers in diameter, and it has a very thick melanin-lined cell wall with very reduced porosity. That enables it to build this enormous hydrostatic turgor. It does that by accumulating huge concentrations of glycerol and also some other polyols in the aprosorium. They generate osmotic pressure. So water is rushing in continually into the cell, but there's no efflux of the solute because the porosity of the cell is reduced by this thick melanin layer, so nothing can actually escape from it, but water can continue to rush in. And these cells form in dewdrops. So you get this enormous turgor build-up. You can see that the aprosorium also sticks tenaciously to the waxy cuticle layer, and in fact it will only form on hard hydrophobic layers. So what we've learned about aprosorium development is there are essentially 3 prerequisites to making one of these cells. And the first of which is this has to be a hard hydrophobic surface. So in the lab we have to use hydrophobic glass or Teflon PTFE membranes, which are very hydrophobic in order to stimulate this developmental process to occur. We know that involves 2 main signaling pathways inside the aprosorium. First of all, the fungus is able to respond to the surface hardness and hydrophobicity by a series of surface-acting proteins, so stretch-activated gated ion channel proteins, and also G-protein-coupled receptors, which are able to respond to ligands released from the leaf cuticle. For example, this one, PTH11. They feed into a mitogen-activated protein kinase cascade. So this MAP kinase cascade has a number of components, and the pivotal MAP kinase, PMK1, is absolutely essential to make an aprosorium. So if you lack this single MAP kinase, then instead of forming this melanized aprosorium, there's just an undifferentiated germ tube. It doesn't make an aprosorium at all. The second pathway that's really important is cyclic AMP-dependent protein kinase A pathway, which is actually responsible for mobilizing many of the storage products from the spore into the aprosorium to generate glycerol, to generate turgor inside the aprosorium. And unless both of those pathways are active, then aprosoria don't form correctly and can't function. The second prerequisite, though, is that this process has to be developmentally tuned in association with a cell cycle. And you can see that most vividly when you look at a 3-celled conidium. So a 3-cell conidium has a single nucleus in each of the cellular compartments. And there's always a single round of mitosis in this cell, which is from which the germ tubes emerge. You can see that single round of mitosis has occurred. So 3 nuclei have now become 4 nuclei. But then what happens is that these 3 nuclei are destined then to be degraded. So the first one went there, this one will go next, and then finally this one. And that always happens. That's completely invariant. So every time there's a rice blast infection, the spore has a single round of mitosis, and then the 3 nuclei that remain in the spore are degraded by autophagy. So that always happens. So 3, 4, 1. And I saw that many, many years ago. Even as a postdoc, I'd noticed that pattern. But at the time, there was very little we could do to understand it. What we now have been able to do is generate a whole series of conditional mutants, so temperature-sensitive mutants, so we could examine those commitment points. And that's enabled us to define some cell cycle checkpoints. There's actually an S-phase-dependent, DNA replication-dependent checkpoint, which is required for initiation of aprosorium development. And then the fungus has to have passed into mitosis in order for the aprosorium to mature. And that mitosis has to be complete in order for there to be cytokinesis and this cell to become separate. And you can see that in this movie, how after the nuclear division has occurred, there's the nucleus moving into the aprosorium, and immediately it triggers this cytokinetic event. So this is an actomyosin contractile ring forming, which occurs directly on transit of that nucleus into the incipient aprosorium. So you can see that this whole process is tightly coupled to cell cycle progression. And if you block those cell cycle progression points, then the aprosorium doesn't form properly. And the last thing that's required is the 3 nuclei have to degrade. So though you saw in the movie how those 3 nuclei degrade, well, this is where we've blocked that process. So in the control, we've just got a single nucleus inside the aprosorium. But in this mutant, we've actually deleted a gene involved in autophagy, so it's ATG8, which is involved in making the autophagosome. And you can see now we've got 3 nuclei still inside the spore. They're distended because there's been a sort of an attempt to degrade them, but they're unable to degrade in the absence of the autophagy machinery. And that means the aprosorium that forms, although it melanizes, it can never re-establish polarity. So it's unable to bring about plant infection. It renders the fungus completely non-pathogenic. So those are the 3 prerequisites you need to make an aprosorium. You have to undergo its cell cycle control, surface sensing, and an autophagic cell death in the spore. What happens then is the aprosorium, having developed this enormous turgor pressure, it will generate a rigid penetration peg just at this point in the aprosorial pore, where there's an absence of the melanin layer. There's a very thin cell wall layer in contact with the rice leaf surface. And then what you see at the base of the aprosorium is this severe membrane curvature generation, and then formation of this rigid penetration hypha, which will rupture the cuticle. And the fungus will rupture plastic membranes in the lab. So we know it's although in the context of a leaf, it actually secretes methyl esterases, it secretes cutinases at that point too, it's able to break plastic and inert surfaces. So it's the force generation that's responsible for the fungus being able to colonize tissue. So how is that protrusive force generation generated? Well, we know that there's actin remodeling occurs at the base of the aprosorium. So this is a big actin ring, a toroidal structure, a donut-shaped structure that forms just at the base of the aprosorium. And that occurs during aprosorial maturation. And that's scaffolded by a set of proteins which are called septins. So septins are GTPases. They're another component of the cytoskeleton. They form heterooligomeric arrangements, first discovered in yeast associated with cytokinesis, associated with budding, but present in mammals, present really throughout the metazoa, but absent in plants. And what's interesting about them is that they're able to form these gauzes, collars, rods-type structures, normally at plasma membrane sites. And they can act as lateral diffusion barriers, so they can hold certain proteins in a particular domain, so they can prevent lateral diffusion in a membrane. And they can also scaffold actin and act to rigidify the cortex. So there's a big cell, a big annulus or ring structure at the base of the aprosorium, which is formed of 4 septins, form this heterooligomer. And that scaffolds actin, rigidifies the cortex, but also acts to maintain polarity determinants, proteins involved in actin polymerization and involved in membrane curvature generation in the center of the aprosorium associated with this production of this penetration hypha. And you can see that in 3D. If we spin that aprosorium, you can see there's the septin ring right at the interface between the fungus and the rice epidermal layer. So we wanted to understand this process and try and work out, well, how does the cell generate pressure, first of all, which we knew the biochemical mechanism through glycerol generation, but how is the decision made that a certain threshold of turgor has been met such that the fungus will then repolarize? So to do that, we actually took a mathematical approach. We talked with some mathematical modelers and we described the process in detail. We then undertook a lot of measurements of the geometry of aprosoria, the relative concentrations of cellular constituents, and talked to them really about the positioning of melanization and this sort of reciprocal arrangement between the absence of melanization and the production of septins and remodeling of actin at the base of the aprosorium. And on the basis of that mathematical model, we were able to simulate aprosorium penetration. And this is a simulation of the mathematical model. You can see that the aprosorium in this model swells, and because of the absence of melanin, so melanin is present in the dome of the aprosorium, but absence at the base, at the aprosorial pore, which mirrors what we see in the lab, at that point we actually see septin accumulation in this annulus leading to actin remodeling and then rapid actin polymerization at the base of the aprosorium, leading ultimately to force generation, and so protrusive force generation. And this is a geometric evolution law model which we can then simulate using this animation. Well, one of the predictions of the model is that there had to be a turgor sensing mechanism, and because the turgor sensing mechanism led to septin recruitment, then it would be likely that the turgor sensor would localize at the point of protrusive force generation, and there we'd see a gradient of turgor sensor forming at that point. And we also were able to model what would happen if we knocked that gene encoding a turgor sensor out. What would be the consequence of that? So we can simulate a turgor sensor mutant. So the absence now, we've got no turgor sensor here. Well, under those circumstances, the prediction of the model is that the turgor will just keep increasing and keep increasing. And it will increase to a point where ultimately the cells would simply rupture without ever undergoing repolarization. So we had some characteristics of a turgor sensing mechanism. So on the basis of that, we then went searching for a turgor sensor. And what we found was a protein called SLN1, which is a histidine aspartate kinase. When we localized it, we found it was first of all in the cortex of the aprosorium, but then as the aprosorium matures, we see increasing concentrations of it at the point of repolarization associated with where the septins and actin accumulate. So sharing some of the characteristics, when we deleted the gene, what we found was runaway pressure. We just saw runaway turgor and no repolarization. So we saw cells continually generating pressure, but they were unable to ever repolarize. So it shared some of the characteristics, again, that the model had predicted. And you can see that the movement of the aprosorial turgor sensing kinase, SLN1, this histidine aspartate kinase, as the cells mature, more and more of it is present at the base of the aprosorium. You see this line scan showing it's actually in the center at the base of the aprosorium. But however, if we artificially lower the turgor of those cells, if you put them in hyperosmotic concentrations of glycerol, so you lower their pressure, then that movement to the center doesn't occur. So there's actually a pressure-dependent element to the localization of this sensor kinase at the base of the aprosorium, so, which is, again, consistent with that. When we look at the role of the septins in terms of scaffolding actin and all of those processes at the base of the aprosorium, so here's our septin ring and actin ring. So you can see that these are two different septins. They both form this heterooligomeric ring structure at the base of the aprosorium. And then, and you can see actin forming in the same way. This is an actin binding protein, gelsilin. This is a kinase associated with septin activity. And these proteins are all involved in repolarization, either polarized exocytosis through elements of the exocyst complex, T1 involved in actin polymerization, and RVS167 involved in endocytic activity. So all of these processes involved ultimately in the aprosorial repolarization process. But when we take out our putative sensor kinase, then we can see they're all mislocalized. So the septins don't recruit properly, and we see none of that organization of those structures at the base of the aprosorium. And what that's enabled us to do is to put together a model for how we think the aprosorium is functioning. And a lot of the work in my lab now is testing this model to try and test some of the assumptions of how we think an aprosorium makes this transition from turgor generation through to repolarized growth. So first of all, we know that the fungus is generating this enormous pressure inside the aprosorium, and that's due to a combination of glycerol biosynthesis and also melanin biosynthesis in the cell wall. through the activity of the cyclic AMP pathway and the PMK1 MAP kinase pathway. Once a threshold is actually reached, then SLN1 we know interacts with 3 stretch-activated gated ion channel proteins. We know that by co-immunoprecipitation analysis, and also a glycerol efflux pump, MIP1. And we also know that it physically interacts with elements of the cell integrity pathway, particularly protein kinase C, which directly phosphorylates part of the NADPH oxidase complex NOXR, a regulatory subunit. That leads to a burst of reactive oxygen species and recruitment of a polarity determinant CDC42, and ultimately to a whole set of different processes in the base of the aprosorium. So septin recruitment and septin ring formation, and then targeted exocytosis, rapid cell wall biogenesis, chitin and glucan synthesis at the base of the aprosorium, and this rapid actin polymerization to lead to that protrusive force generation. And ultimately, all of those things lead to PEG generation. But interestingly, only once a cell cycle control point is being triggered. So again, the nucleus in the aprosorium has to have passed into S phase to enable any of this to occur. So it's also controlled by a cell cycle checkpoint. So we're beginning to put together the different parts of the puzzle, and what we're now doing is systematically working through some of those assumptions and finding more evidence for the way in which this signaling pathway ultimately leads to PEG formation and an aprosorium cuticle rupture. So that's all the things that we know about so far that are happening on the leaf surface, and now we're going to move inside the plant. One of the things I used to always get at conferences were lots of people used to say, when are you ever going to start working on the plant? Because we spent so much time working on aprosorial determination and formation because it was so interesting. But we've now started working in the last few years about trying to understand what happens next. What happens when the fungus is actually occupying and invading plant tissue? What are the changes that it undergoes then? And in many ways, it's no less remarkable from a developmental biology standpoint. So how does the fungus then invade plant tissue? Well, if we lift the aprosorium and have a look underneath in a confocal image of a plant infection, you can see that rigid penetration hypha has ruptured the cuticle and has now entered the underlying epidermal cell. So this is the rigid penetration hypha that's actually ruptured through the cuticle. But what it does when it does that is it will then immediately differentiate into a different growth form. And this is these bulbous branched hyphae. And they actually divide by a rather different mechanism than when the fungus is growing in culture. If we grow the fungus on plates, then it will your classical cylindrical hyphae, which undergo branching. Whereas inside a plant cell, they undergo this axial budding. They're much more like a yeast, so they will undergo budding type growth, and you can see these bulbous branched hyphae filling epidermal cells. But remarkably, despite that cuticle rupture, which involves this force generation, the plasma membrane of the plant is not ruptured at all. So what it sets up is actually a very sensitive, rather intimate association between plant and pathogen. So we can see that if we make a rice transgenic line where we've targeted GFP to the plasma membrane, this is it's actually an aquaporin encoding gene, LTI6B. So this is a rice transgenic line. The fungus is just expressing cytoplasmic red fluorescent protein. And what you can see is the plant plasma membrane invaginating around the invading fungus. So the plant plasma membrane is completely intact. So the cell that's being occupied is viable, and the fungus is able to suppress immunity, and we'll talk about that in a moment, in order to occupy a large amount of the volume of the cell. So it's obviously stimulating rapid membrane biogenesis by the plant to occupy this invading fungus. Now, as the fungus then moves from one cell to the next, it's able to maintain that membrane integrity. So often, this is the cell that's been initially infected. There's an aprosorium out of focus on the leaf surface. And as this cell's been vacated, the membrane integrity of the cell that's the fungus is departing from is often lost. But as the fungus moves into the adjacent cell, you can see that it's got the membrane wrapped around it again. And as the fungus moves throughout plant tissue, you'll see this zone at the boundary where the fungus is growing of always intact, viable plant cells. So it's always occupying living plant cells. The cells that it departs often lose viability, and the fungus ultimately will cause necrosis, and that's where you'll get aerial sporulation and the growing of that disease lesion. But every time the fungus is growing into new tissue, it's always moving into living plant tissue. It's always moving in a biotrophic way into plant tissue. So the fungus is undergoing secretion of two different types of effectors. So effectors are proteins produced by pathogens largely to suppress immunity and aid proliferation of the pathogen in plant tissue. Magnaporthe has two different types of effectors, one of which are the ones shown here in green, which are secreted from the hyphal tip, and they occupy the gap between the fungal cell wall and the plant plasma membrane, the apoplast. So those are apoplastic effectors, and they do things like suppress chitin-triggered immunity. They will suppress surface-acting sensors of pathogen presence, so things which are involved in PAMP-triggered immunity, so involved in the immunity that plants trigger when they just respond to the presence of pathogens at their periphery. But meanwhile, the fungus then also secretes effectors which are destined to be delivered inside plant cells, and they will cross the plant plasma membrane in a way that we don't really understand, but they'll enter the plant and they will either target organelles or they'll move into the cytoplasm of rice cells. In the rice blast fungus, we can see them in this structure called the biotrophic interfacial complex, a membrane stack of plant membranes on the outside of the fungus where we can see those effectors accumulate. And if we photobleach that, we can see that it's filling up again. So the fungus is actively secreting effectors through that point into the plant cell. So this focal immune response is then taken advantage of by the fungus in order to deliver effectors into the cytoplasm of the cells that it's occupying. So there are those two different types of effectors. And what's remarkable is there are just so many of them. So Magnaporthe, there are around 400 effectors that have been described in different host-limited forms of the fungus. And many strains will have more than 200 different effectors. They're differentially expressed. So this is a RNA-seq experiment where on two different rice lines where you can see that there's temporal co-regulation of a whole set of effectors which come up here at between 48 and 72 hours after infection. And there's a class of these 91 effectors which are temporally co-regulated, which we call MEP effectors, which we've begun to functionally characterize. And we've actually deleted all 91 of them, and we've been busy trying to work out what their functions are. So to give you an idea of just one of them, because we've been characterizing so many, to give you an idea of the type of things they do, this is an effector called MEP3, which, if you delete the gene, it has an effect on the ability of the fungus to cause disease. There's less disease symptoms. The plant will undergo a de-repression of plant defense. You'll see a reactive oxygen species burst in plant cells when they're infected with the mutant. So, and there's a de-repression of gene expression associated with plant defense. But this is an effector which actually moves to the chloroplast. It's present in a subset of chloroplasts inside the plant, and it binds to part of photosystem 2, a subunit called PSBP. And there it's able to suppress chloroplast-triggered ROS burst, so it's actually able to suppress reactive oxygen species generation in the chloroplast as part of the plant defense response. So these effectors are triggering a whole set of different, suppressing a whole set of different plant defense responses. So the fungus is swamping the plant with those as it's entering new tissue. And the culmination of that process is the fungus has a remarkable ability then to occupy plant tissue very, very rapidly. And this I'm going to show you a time-lapse movie which is taken over 16 hours. And what you'll see is just the manner in which this fungus is able to occupy plant tissue. This is a whole plant. So you imagine we've got a plant in a pot and we've dragged the leaf over onto the stage of a laser confocal microscope, and we're just now going to visualize infection over this time point. So the initial things you'll see are aprostoria and then the initial penetration hyphae. The GFP is an actin binding protein, so you're looking mainly at actomycin, contractile rings, points of septation. You can see the fungus now rapidly moving within cells and then between cells. And over a period of 16 hours, it will completely fill that epidermal layer. So it will completely fill rice tissue very, very rapidly. So you can see these effectors are tremendously effective at suppressing defenses and allowing the fungus to rapidly colonize tissue. If we do look at this in a slightly different way, if we actually now just focus down through an entire leaf, this is not a movie, this is just focusing down through a sort of big Z stack, you can see how the fungus is actually filling epidermal tissue, and then it will move further down into mesophyll tissue, and you'll see longer runner hyphae make their way to the vascular tissue so that it's able to occupy whole leaves and spread into stems and spread systemically throughout plant tissue. So these effectors are really very, very good at enabling the fungus to rapidly invade and occupy plant tissue. But what was intriguing about seeing all these things as movies for the first time was actually trying to then think about, well, how does the fungus traverse from one rice cell to another at the same time as maintaining membrane integrity? So how is it able to accomplish that? As it moves into a new cell, it's surrounded by plant plasma membrane. So we decided to look at that process in more detail. And this is a cropped part of the movie I've just shown you, showing what happens when the fungus moves from one rice cell to the next. So you can see it begins to swell, then you'll see that actomyosin contractile ring, so it's undergoing severe hyphal constriction, and then it's moving through the other side into the adjacent cell. And it's undergoing that swelling, constriction, and moving through every time it moves from one cell to the next. So we decided to look at that in even greater detail. We took the same section, and what we did is then did some transmission EM, and we took many, many sections through this part of the cell wall and then built them up using a tomographic reconstruction. So you can see we're now taking around 100 sections through that region, and then you can build up and look at the extent of hyphal constriction. So you can see what's happening there is that the fungus is constricted to around 300 nanometers or so in diameter in order to move from one rice cell to the next. And that matches the diameter of a pit field, which is an area where plasma desmata, the cytoplasmic continuity between plant cells, where they accumulate, where they cluster. And what we found is that those points of traversal correlate remarkably strongly, closely with the points of where pit fields are present between rice cells. So the fungus is utilizing pit fields in order to secure its movement from one rice cell to the next. And you can see that here. This is a pit field which has been located by the fungus. It's moved through that region. You can see those hyphal constrictions again shown here. The other thing that we can tell is that Magnaporthe is able to manipulate plasmodesmatal conductance. So we know that it's able to do that because if we take one of those effectors, which we know is delivered into a rice cell, and we actually hooked a nuclear localization signal onto it, so we can then send it to the plant nucleus. And that's this effector called PWL2, which we've hooked an NLS and an M-cherry fluorescent tag onto it. And you can see this effector has now made its way to the plant nucleus. But not only is it in the plant nucleus in the cell that the fungus is occupying, but it's in this one, and it's actually at least 5-6 cells away. It will diffuse into those cells and it will move into those nuclei. So clearly those plasmodesmata are open and the fungus is able to secrete effectors which will then cause effects in adjacent cells. And we've seen many, many instances of this where the fungus is able to manipulate behavior in cells well away from where it's actually infecting. So clearly it can open plasmodesmata. And we've actually directly measured plasmodesmatal conductance, and we can show that during infection, plasmodesmata are manipulated by the fungus. We can see also something about the mechanism by which that happens, because there's a group of effectors which will localize at those crossing points. So this is an effector called BAS2. We don't know its function, but what we do know is it accumulates at crossing points. It accumulates at pit fields and those sites of hyphal constriction. So the fungus is clearly able to manipulate plasmodesmatal immunity, and it's using those sites as its means to traverse from one rice cell to the next. So the problem is, how do you study this? So how can you study the process of cell-to-cell movement? Because what we really need is a way of conditionally blocking this cell-to-cell movement process. So to do that, we went back to a pathway which we were really familiar with, but from another context. So the pathway I told you about at the beginning, this pathway, the PMK1 pathway. So remember I told you that PMK1 is absolutely essential for aprosorial formation. So if you delete this single MAP kinase, then you can't make an aprosorium, you just get an undifferentiated germ tube. But I remembered that actually if you take a PMK1 mutant and you just take spores or hyphae and you inoculate plants directly, or you inoculate wounded plants, or you just simply inject that fungal material directly into plant cells, then they never cause disease symptoms. This mutant is completely non-pathogenic, completely unable to cause disease. So clearly, as well as affecting aprosorium development, it must be doing something else. It must be doing other things after infection. And we thought, is there a way that we could potentially study that? So we decided to take a chemical genetic approach. And the way we did that is we made a point mutation, this glutamine to glycine mutation in the gatekeeper residue, which is in the ATP binding pocket of that MAP kinase. We were then able to take that mutant allele and then do a knock-in, so we do an allelic replacement of the native gene locus back in the fungus. So we've now made a mutant where we've got a single point mutation in the ATP binding pocket. That will make that kinase then susceptible to an ATP analog called naphthol PP1. So we can make an analog sensitive kinase, which means that we can now switch the kinase off whenever we want to using a drug. So we've now got a conditional mutant. So to test that that worked, first of all, we took the mutant phenotype we were familiar with. So this is the PMK1AS analog sensitive strain. And you can see normally in the absence of an inhibitor, it makes aprosauria normally. But if you add the inhibitor, it stops making an aprosorium. So that showed that it was behaving in the predicted way, and we could test its kinase activity and do all the biochemical checks, and that showed that we'd actually been able to eliminate that single kinase. It was the only kinase affected by the drug treatment. So then we could do the experiment we'd always wanted to do, which is allow the fungus to infect and then switch it off. So first of all, this is what happens in the analog sensitive kinase mutant in the absence of the drugs. There's no drug. This is the same as the wild type would be. So this is the analog sensitive kinase mutant with no drug present, and you can see that it will fill those epidermal cells, and it fills them until it gets to a point, and then it breaks out. You can see it undergoes a hyphal constriction. It's breaking out into all the adjacent cells. And it's a remarkably synchronized event, and we see this time and time again. So if we simply add inoculate the wild type or this analog sensitive mutant in the absence of the inhibitor, then it will fill epidermal cells, and then you'll get this, they'll locate pit field sites and then break out into adjacent cells. So then we decided, now let's add the inhibitor. So this is the experiment where we added the inhibitor. So now we've got the same strain, but we've added the kinase inhibitor. And you can see it filling that epidermal cell and filling it, and it never gets out. It can't escape at all from that cell. It's completely trapped. So it's unable to move through those pit field sites into the adjacent rice tissue. It's totally trapped within that cell. And you can see that shown here. Here's the fungus. It seems to be scanning the cortex. It's scanning the periphery of the cell, but it can't undergo that movement through into adjacent rice cells. It becomes completely trapped. So what's happening? Why is that occurring? How is that single MAP kinase? What are the effects it's having to bring about that process? So we know that plasmodesmatal immunity is triggered by pathogen perception, triggering a ROS burst, a reactive oxygen species burst, and then release of salicylate, ultimately leading to callosynthase and then occlusion. So callosynthase activity will then form callose and occlude those, shut the plasmodesmata, so sealing pathogens in a single cell. So that's what would happen. So we wondered, well, what's happening to those immune responses when we've blocked cell-to-cell movement? So we decided to look at that process. So in the absence of the inhibitor, the fungus is in this cell, and it's broken out into the adjacent cells. And the plant seems to be completely oblivious to this. We don't see any expression of plant defense genes, and we don't see any reactive oxygen species burst. This is diamidinobenzidine, so DAB staining, to look at reactive oxygen species generation. If we add the inhibitor, you can see the plant is now perceiving the pathogen. So it's now perceived the pathogen as being present there, and it's now sealed in this area where there's this ROS burst has been triggered. We can also see the effect on callosynthesis. So normally in the absence of the inhibitor, the fungus is able to, so here's the fungus here growing out. In the absence of the inhibitor, it's growing into these adjacent cells. And as it moves from one cell to the next, the plant actually responds rather late and forms a callose collar around the hyphae that's already left. So it responds very late to the fact that the fungus has actually already moved from one cell to the next. And you see these callose collars, which are sort of footprints of where the fungus has already been as it's moving through, but there's no rapid response. Whereas if we in this case now we've added the inhibitor. We've got 3 different infection points here. This cell's infected, this one, but they can't get into these adjacent cells. They're trapped because of the inhibitor. And what you can see is those cells, the adjacent cells, have responded by callose occlusion at all of their pit field sites. So by the fact that this fungus is trapped, the plant is now able to respond, triggers its defenses, and is perceiving the pathogen as being present in each of those cells, and it's unable to move from one cell to the next. So what about those effectors that we know localize to crossing points? So this is one of those fungal effectors. So this is BAS2, and we know it's normally at these crossing points and also present in the biotrophic interfacial complex in cells that are being occupied. So this is in the PMK1-AS strain, again, in the absence of the inhibitor. This is what would normally happen in the wild type. So the effector is localized into those crossing points. If we add the inhibitor, we don't see any localization of BAS2 to those crossing points, but also we don't see any expression of the gene. So it's not actually even being expressed. So it shows that those effectors, which would normally go to pit field sites, are under control, ultimately, of the PMK1 MAP kinase cascade. So we can look at that in much greater detail now, so we can do an RNA-seq experiment. So this is where we've just simply infected rice at 26 hours and either added the inhibitor or not added the inhibitor, then extracted plant tissue, extracted RNA and done RNA-seq analysis. You can see there are more than 700 genes that are differentially expressed during that point. 212 of those encode secreted proteins, many of which are temporally co-regulated with a group of effectors which we know are involved in plant defense suppression, and 10 of those were ones which we'd actually already characterized. So we know that so there's a subset of effectors which are regulated as a consequence of this pathway being triggered. So clearly the fungus is suppressing plasmodesmatal immunity as it's moving from one cell to the next. But actually that's not all it's doing, and in fact that's probably even not the most important thing it's doing, because the other thing that actually has to happen is the fungus has to undergo this severe hyphal constriction. And that brings us back to the class of proteins which we were looking at in the context of aprosorium development, which are the septins. Because what we found is at the point of crossing, you can see there's this septin collar forming as the fungus is moving from one cell to the next. So septin collars always form at those points. And if we then look at the same crossing points, either in the presence or the absence of the inhibitor, we can look at the effect that the absence of that MAP kinase can have on septin deposition by doing that. And that's this experiment. So you can see this is the absence of the inhibitor. Again, we've got these beautiful septin collars. Every time the fungus is moving through a pit field, there's a septin collar, which is showing the degree of hyphal constriction as it's moving, this hourglass shape, as it's moving from one cell to the next. However, when we block that process, then there's a loose, diffuse localization of septins, and they don't ever form those collar-shaped structures at the aprosorial, at the boundary between the fungus moving from one cell to the next. And we also now know, and this is rather difficult to do, so we've now made some temperature-sensitive conditional septin mutants, and we've done that using CRISPR mutagenesis. We've actually now got triple mutants because there are 4 septins. So we've got triple mutants where we've got them all under control of a temperature-sensitive switch so that we can then conditionally inactivate the septins. And our preliminary evidence of that, and some evidence we've got, which we published from looking at knockout mutants, suggests that this process, again, is septin-mediated. So just like the aprosorium, in order to move from one cell to the next, the hyphal constriction is absolutely pivotal, and it's a septin-dependent process. We also know that if you simply suppress plant immunity, then the fungus is still trapped. And the fungus never actually gets out of that cell, even if you completely suppress immunity. And in fact, even if you kill plant tissue, the fungus remains trapped within those cells. So it really shows how important the hyphal morphogenesis aspect of this is to moving from one cell to the next. And finally, just to sort of put that into context, in a way, our work has come full circle, because we spent a lot of time working on the aprosorium and this specific, very specialized, cell cycle controlled, orchestrated process by which a pressure generating cell is able to undergo a switch from anisotropic to isotropic growth in order to rupture the cuticle and enter rice cells. Actually, when you look at the boundary between one rice cell and the next, there are some remarkable parallels emerging in the way that those cells actually work. And if we've begun to now use our tomographic reconstructions to try and map out what's actually happening at those crossing points. And I'll just show you one of those animations now. So this is, again, looking through a single crossing point. So one of these crossing points here, we've just taken this and done some EM analysis on it. And you can see, if we look through there, you can see the degree of hyphal constriction. And then we're now actually mapping each of the cell layers. So we've mapped the plant cell wall layer, the fungal cell wall layer, and now we can start mapping organelles. And what we can see at each of those crossing points is a rather uniform set of structures, a pair of peroxisomes flanking the site of hyphal constriction, this ER continuity. We can see mitochondrial clustering as well. So what we're beginning to think of is this is not simply a cell-to-cell traversal mechanism, but this is much more in parallel with the aprosorium. This is actually a specialized developmental process. And we know that, as I said, even in inert substrates, it has to undergo this mechanism to move from one cell to the next. And if you go back through the literature, some keen-eyed scientists more than 50 years ago coined the term transpressorium, because they said you can see hyphal swellings as the fungus is moving from one cell to the next, and it's analogous to the aprosorium. And I think they're right, that this has actually got some remarkable parallels. Although these cells don't melanize, and we don't see this massive turgor generation that's necessary to rupture the cuticle, there certainly seems to be developmental parallels, and certainly at the molecular level, many of the components are conserved between the mechanism by which the fungus moves from one cell to the next and the way it actually enters the rice leaf initially. And I think there are probably some interesting evolutionary questions there about how this whole process emerged. So the model for what's actually happening in the base of the aprosorium is that this PMK1 MAP kinase cascade is triggering effector gene expression, suppressing host defense and plasmodesmatal immunity. But also, equally important, it's undergoing this host sensing mechanism leading to this rapid cytoskeletal remodeling, severe hyphal constriction, septin deposition, and ultimately cell-to-cell traversal. So just to summarize everything I've told you, I hope I've left you thinking that the developmental biology of these plant pathogenic fungi is pretty interesting and pretty neat in terms of the developmental processes and transitions they can go through. And also the fact that infection is not a random process at all. It's highly orchestrated and highly regulated and always linked to cell cycle progression. And we can actually see that in Plantar too, with some of the more recent work we've done. But certainly in terms of aprosorium formation, we know that's triggered to those three prerequisites that are required for it to occur, including responding to a surface, cell cycle progression through S phase, and then autophagic cell death. And then once the fungus is actually inside the plant, having gone through the septin-mediated cuticle rupture process, then it's able to deploy a whole battery of fungal effectors. They suppress immunity, and cell-to-cell movement involves this specific wall crossing process, which is directly analogous to the way the aprosorium works. And this term, the transpressorium, may be something that we'll adopt in future in terms of trying to understand how it works. So the people who are involved in doing this work, much of the work I've told you about was carried out by Lauren Ryder, who did all of the work on the turgor sensing kinase, but particularly working with our mathematical modeler collaborators at the University of Sussex, Anatida Madsva-Muse and Chandrasekhar and Vanessa, who did all of that mathematical modeling work. The bulk of the rest of the work is all really done by the people on this slide. So Clara was involved in effector secretion, Bozeng in effector regulation, Vincent was involved in effector characterization with Shah-Yan, Neftali is now doing phosphoproteomic analysis downstream of that wall-crossing mutant, and Andy and Alice are involved also in aprosorial and septin interactions, and Miriam was involved in all of the cell cycle work. Most of the work I've told you about was funded by the ERC, and now through the Gatsby Charitable Foundation and the Helping Scholars Program, now that I've moved to the Sainsbury lab. But much of this work started when I was at the University of Exeter with many of my great collaborators there too. So thank you very much. As I say, it's been a great honor. Thank you.
Thank you very much, Nick. We've had 3 plenaries and 3 very different plenaries. And it really illustrates the diversity of the society, which is really one of its strengths. And here in Nick's talk, an in-depth talk about elucidating a host-pathogen relationship or dynamics. But I think it was the beginning message that really got me, and that it helps to us address a global challenge, and that is food security. So Nick, thank you very, very much for your presentation. We have something small in honour. And thank you very much. Thank you very much. Thank you. Okay, so please hang around, because we also have another important event happening, and that is to acknowledge our early career researchers, our young scientists, and we have a number of awards that we will be handing out. So I'm going to run through these and ask the people that I will be mentioning to come up and accept their awards. We'll start off with the President's Medalists. These are early career scientists within 10 years of receiving their PhD that have shown outstanding merit in their research, underpinned by leadership and creativity. So for the animal biology section, Sonia Au, can you please come up and accept your medal? The President's Medalist for the plant biology section is Daniel Gibbs. And the President's Medalist for the cell section is Ian Johnston. Thank goodness this lecture theatre isn't any bigger, because otherwise you wouldn't be getting lunch. Okay, so moving on, we are going to award the Young Scientist Award winners. And the first place goes to Michael Sackville from the University of British Columbia. And we've got somebody running. Good on you. And we have two runner-ups, Lena Thorat and Charlotte Lemuel. Can you both please come up? Thank you. Young Scientist Award for Plant and Cell. First winner is Thomas Challoner from the University of Exeter. And the runner-up is Christopher Hepworth, and a second runner-up, Sophie Harrington. And finally, we have the Irene Manton poster prizes. So I'll call out all three winners for the plant, animal and cell section. For the plant section, Emily Harrison. For the animal section, Tiffany Armstrong. And for the cell section, Zahara Razegi Mohagan. And this is one of those situations where I feel I've been stood up. So just to draw this to a close, the Society really does want to create opportunities for our early career researchers, our PhD students, etc., and postdocs. And if you can think of what opportunities you would like, what we could offer, please offer us some suggestions. We have a suggestion box up at the reception. Please just write a note down. We welcome your ideas as to how we can improve things and create more opportunities for science. And we have, who do we have here? We have Emily. Good on you, Emily. Well, folks, I think we can break for lunch. Thank you very much for your attention. Thank you to the 3 plenary speakers. Enjoy your lunch. Thank you.